So in this lecture, we're going to take a look at specific parts of the bacterial cell, and we're going to start by looking at the cell wall. In nearly all bacteria, there is a cell wall that surrounds the cell membrane. And sometimes there's even an additional outer layer beyond that. So that is sort of the outermost area. Inside of the cell, we see the cytoplasm that includes organelles such as ribosomes, and then there's a nuclear region, sometimes referred to as the nucleoid here. And then on the external surface of this bacteria, we find external structures like the capsule that surrounds the cell wall and the cell membrane, or flagella or pili even. So flagella are organelles that are used for locomotion, and pili are used for attaching to other things. But where we want to really put our focus right now is on the cell wall and the cell membrane. So the cell wall is external to the cell membrane, and its primary function is to maintain the shape of the cell, to make sure that the cell is able to withstand any osmotic stress and to hold and protect the cell membrane, which is a much, much weaker structure. It also provides anchor points for structures like flagella, and because of the porosity of it, it's very porous, it doesn't have any role really in regulating what can go into or out of the cell. That is a function that is provided, or that's limited really to the cell membrane itself. The cell wall is built of a molecule called peptidoglycan, and that's the primary component of the cell wall. It's sometimes referred to as murine as well. But we will, in this class, we'll only refer to it as peptidoglycan. This is going to be our primary way of talking about this molecule. And it is a very, very, very large molecule. It's a polymer of a number of sugars, and the sugars have attached to them some amino acid groups, or I'm sorry, not amino acids, but just amino groups. Amino groups are in NH2 that's attached to a carbon. And then they are cross-linked by a polypeptide that's built of 4 amino acids. And so you can think of the structure of the cell wall as almost like a bunch of layers of a chain link fence. So the chain link is built of the peptidoglycan and the amino groups, and the layers of chain link fencing then are held together by these 4 amino acid polypeptides. And in gram-positive cells, the cell membrane is much thicker, so there might be as many as 40 of these layers of chain link fencing around the bacteria if it's gram-positive. In a gram-negative bacterium, it is much, much thinner. So let's talk about the cell walls then in gram-positive and gram-negative cells. So in gram-positive cells, we also find included in this massively thick layer of peptidoglycan the amino acid lysine and another acidic organic molecule called teichoic acid. And like we said, it is a very, very thick layer. And here, looking at this electron micrograph, that outermost layer there is built of the peptidoglycan, and then here is where the cell membrane is. So you can see the thickness that we associate there with the cell wall. And there it is. It's 40 layers, and we've got these kind of pinky colored things. Those are the teichoic acid that are also in there, embedded in the peptidoglycan, kind of holding things together. Gram-negative cell walls, though, rather than having lysine and teichoic acid, they include diaminopimelic acid, another one of the amino acids. And we see that the cell wall in gram-negative cells is very, very thin in comparison. So here's that peptidoglycan layer in a gram-negative cell wall. The cell wall in gram-negative bacteria really helps to protect the bacterium from host defenses, because the amino acids that are found in the polypeptides of the gram-negative cell wall, they are mirror images to the usual forms. We talk about these mirror images as stereoisomers. So an isomer of a molecule has exactly the same construction, but it's, I'm sorry, let me try that again. It has exactly the same atoms involved in the molecule, but they're put together in a different way. That's an isomer. And when it's a stereoisomer, that's when you see these mirror images. And so the cell wall is super protective of health, from the host defenses, because most hosts can't digest the peptidoglycan in cell walls. But it is possible to overcome cell wall defenses. One way is with antibiotics like penicillin and other antibiotics that interfere with the peptidoglycan synthesis for the cell wall. And so by interfering with the synthesis of peptidoglycan, structural weaknesses are found in the cell wall, and those weaknesses then allow osmotic stress to burst the cell, and the cell dies then. So that's how penicillin works. We'll talk more about that. We've got a chapter coming up later on where we start looking at chemotherapies and antibiotics and how they work. So more to be coming on that later. Humans produce an enzyme in our tears. It's found in tears and some other human secretions, but we think of lysozyme, this enzyme, being primarily in the fluid that comes from the eyes. So lysozyme is important because it is one of the few enzymes that is capable of digesting peptidoglycan. And so we are able to avoid the eyes as a portal of entry for many bacteria because of the lysozyme that is found there. So both of these kinds of defenses against the cell wall create weaknesses so that osmotic stress will burst the cell and kill it. Okay, so in gram-negative bacteria, we also see an outer membrane. So we're talking here now about the outer membrane. So it's a membrane that is on the external surface of the peptidoglycan cell wall. It's bonded, so it is structurally bound to the peptidoglycan. And unlike the cell membrane, the outer membrane has very little function in controlling movement of materials into or out of the cell. But it does have special proteins, they're called porins, in that outer membrane that produce channels that allow molecules to pass through the outer membrane very easily. But it does prevent penicillin from being incorporated into the cell. And so many gram-negative bacteria, because of this outer membrane, are resistant to penicillin naturally. So it's not an acquired resistance that's evolved over the past, how long has it been since we've had penicillin? 80 years or so. Instead, it is a natural, it was in the bacteria before we started treating people with penicillin. So also associated with the outer membrane is a material that's referred to either as endotoxin or as lipopolysaccharide. So lipopolysaccharide or endotoxin. And lipopolysaccharide is a big mouthful to say, so LPS is frequently used to refer to this molecule. So all three terms mean the same thing. I'm sorry about that. So lipopolysaccharide, or endotoxin, is found in the outer membrane that's associated with gram-negative bacteria. And as the bacteria are killed, or as they die, just naturally, then this lipopolysaccharide material is released from the membrane. And when it gets into the blood and then distributes through the body, it can cause a number of very, very dire outcomes, especially if it's in high concentration in the body. First off, it's a pyrogen, so it causes fever. Pyrogens, by the way, I'll spell that for you. Pyrogen. A pyrogen is any molecule that can trigger fever that talks with the hypothalamus and forces it to reset the body temperature to a higher level. So pyrogens cause fever, and endotoxin is a pyrogen. So it can cause fever and cause blood vessels to dilate, which then leads to a great drop in blood pressure. As we open up all those blood vessels, then the increased capacity of the circulatory system leads to a blood pressure drop, which is what we call endotoxic shock. So all of these, if there's a large number of dying gram-negative bacteria, they'll release endotoxin in sufficient amounts that it can even kill the patient. So it's especially important to know when you're dealing with a systemic infection that's being caused by a gram-negative bacteria, because treating it vigorously can cause a great big release of endotoxin and the resulting endotoxic shock. And so as well, we can use the presence of endotoxin in the body, in body fluids, as a means of identifying that gram-negative bacterial infection is taking place. So in this image here of the cell wall of E. coli, which is a gram-negative bacterium, it's a gram-negative rod. Here we see the inner membrane. Here we see the periplasm with, right here would be where the peptidoglycan is. And then here's the outer membrane with all of these molecules of lipopolysaccharide extending from the outer membrane there. And here are some of the pore channels that allow materials to get into that periplasmic space. Okay, so gram-positive bacteria, on the other hand, we've talked a little bit now about gram-negative bacteria. Gram-positive bacteria have a very thick peptidoglycan layer, as we've said. It can be up to 90% of the cell wall. And there's very little protein associated with this peptidoglycan. It's mostly the polysaccharide layer. If you remove the peptidoglycan from a gram-positive bacteria, then the cell becomes what is referred to as a protoplast. Protoplasts are roughly circular or spherical shaped cell membranes. Even if, say, we had a gram-positive rod, when you remove that peptidoglycan layer, that supporting layer, then the protoplast will be a roughly circular or spherical cell. And the cell will shrivel or burst if it's subjected to osmotic stress in this protoplast state. And so protoplasts, when they're made, must be kept in isotonic solutions. And so they're used as a research tool for folks who are trying to understand how the membrane functions, and with the goal of removing the cell wall so it's not contributing to any membrane function at all. As well, some other characteristics about it is that thick cell wall, if it's a young bacterium, 24 hours old or younger, they tend to retain dyes very well. For instance, in the gram staining procedure, you use crystal violet and iodine as the two dyes that are going to indicate that a gram-positive bacteria is there, because the crystal violet can penetrate and stick to the peptidoglycan layer And then when you add iodine to it, it makes that crystal violet molecule even larger and really locks it in place. In older bacteria, though, older than 24 hours, that cell wall is not near as consistent in how it behaves with dyes like crystal violet. And so it's possible that even though it's a gram-positive bacteria, that when treated to a gram stain procedure, when you rinse away that first stain, the crystal violet stain, and then when you rinse that away from any of the gram-negative bacteria, it may also be rinsed away from older gram-positive bacteria. So the older bacteria tend to either appear gram-negative because the primary stain washes out and only the counter stain remains, or gram-variable with some of the cells retaining the primary stain and other cells losing it. Okay, so with some more stuff about the cell wall here. With a gram-positive bacteria, there is no outer membrane. So it's just the outermost layer of the cell wall is that peptidoglycan layer. And it has a periplasm, so there is some fluid that is found in between the cell membrane and the cell wall, but the space is very, very thin or almost non-existent. Okay, so distinguishing the gram-negative bacteria. As we said, the cell wall in gram-negative bacteria is much thinner, but it's also more complex because of all of this membrane, the outer membrane that is associated with gram-negative bacteria. So about 10 to 20% of the cell wall in gram-negative bacteria is peptidoglycan, and all the rest is other polysaccharides, proteins, and then the lipids of the outer membrane. There's a much larger periplasmic space here, and there's so much larger periplasmic space, and it is a very metabolically active space. The periplasmic space in gram-negative bacteria contains toxins and enzymes that can serve as a defense. So if something tries to come and munch on the bacteria, then if they get through that external membrane, they will release the toxins and the enzymes and be damaged themselves. So if you remove the peptidoglycan from the bacteria, so that in the same kind of a procedure as we did with our gram-positive bacteria, the bacteria becomes a spheroplast that has both an inner and outer membrane, just without this thin layer of peptidoglycan in between them. And the outer membrane provides additional support, so unlike with the protoplast, the spheroplast is much less likely to burst because of that extra support that the external membrane provides, the outer membrane provides. So, bottom line, this relatively little peptidoglycan and the large quantities of the lipoproteins and lipopolysaccharides that we find associated with the cell wall means that the gram-negative bacteria tend not to retain crystal violet iodine when done with the gram stain. When you go and rinse them with alcohol, the crystal violet rinses away, and so it's only the counter stain then that stains a gram-negative bacteria, and they will wind up appearing pink. So rather than being the dark purple that we associate with gram-positive bacteria, we see pink cells instead when we look at them under the microscope. All right, so that's gram-negative and gram-positive. Now let's talk about one last group of bacteria that can be distinguished based on the cell wall differences. And those are the acid-fast bacteria, also known as the mycobacteria. So they all belong together in one evolutionary evolutionarily cohesive group. So they're all much more closely related to each other than they are to other bacteria, these acid-fast mycobacteria. So they also have a thick cell wall. About 60% of it is lipid, though, so that means that there's less peptidoglycan in acid-fast bacteria than there is in a gram-positive bacteria. When you subject them to the gram staining, though, they do stain as gram-positive. But if you use that acid-fast staining technique, the carbylfusion will bind in the cytoplasm, and it really resists being removed by the acid and alcohol decolorizer. So with the acid-fast bacteria staining procedure, that Zeil-Nielsen procedure, the primary dye that's used is carbylfusion, and then it's washed not just with alcohol, but alcohol mixed with an acid. And so carbylfusion will stay in the cytoplasm of the acid-fast bacteria. It doesn't, so it penetrates through the cell wall and gets into the cytoplasm, and then the cell wall protects it from the alcohol and acid decolorizer. The mycobacteria, these acid-fast bacteria, some other characteristics about them, they tend to grow slowly, and that's because the lipid-rich nature of the cell wall prevents, or makes it more difficult, I should say, probably. It makes it more difficult to get nutrients transported into the cell through that very lipid, fatty cell wall. And so that is one reason why they don't tend to grow very quickly. The other reason is when you're making something as expensive as a lipid-rich cell wall, that takes a lot of energy to do. So in general, the acid-fast bacteria, these mycobacteria, grow more slowly than other bacterial types. The genus Mycoplasma, is one bacteria that doesn't have a cell wall at all, which is an interesting thing. So these are wall-deficient bacteria, these bacteria that don't have a cell wall. And so they still require reinforcement, though, to prevent osmotic stress to their membrane. And without a cell wall, then they would be in danger of osmotic stress and bursting. But cholesterol is found amongst the phospholipids of the cell membrane in Mycoplasma, and that helps to stabilize the cell wall or cell membrane, and prevent it from bursting due to osmotic stress. Mycoplasma never has a cell wall. There are some other bacteria that will spontaneously lose their cell wall, either naturally or in response to chemical treatment. These L forms, named for the Lister Institute, where they were discovered, these L forms are very difficult bacteria to treat, and so they can cause chronic or recurrent diseases. So if the bacterium will naturally form wall-less L-form bacteria, then when we give a person, say, penicillin to treat the disease, then it'll kill off the bacteria that happen to have a cell wall, but it will leave the wall-less L-form bacteria alive. And so maybe, there are in general, there are usually fewer L forms than there are regular walled bacteria, and so you can even believe that you have treated the disease and it's been successful, so that you stop the treatment. But as soon as the treatment stops, these L forms will continue to reproduce, and some of their offspring will return to making cell walls, and they can regrow into an infecting population again. So, interestingly, Crohn's disease is one of the diseases that's associated with bacteria that produce L forms. So here we see a normal Bacillus subtilis. So we're using, we have, I forget which species of Bacillus we have, but we've been looking at Bacillus. Oh, now I can't remember. But we've been looking at a Bacillus species in lab. This is Bacillus subtilis with its cell wall, but it can also become an L form, and here we see the more spheroplast looking, or spheroplast or protoplast looking forms of the cell walls. Of these cell wall-less bacteria.